Okay. A little better?
Much better, yeah.
Okay. So this is going to be a little awkward. I'm going to be talking on the old laptop that actually works with the mic and do things on the other one, but we'll make it work.
That's fine, but at least we can understand what you are talking about.
Yeah, sounds good.
Yeah, because before you sounded a little bit like a Dalek, you know.
Okay, sorry about that. I was using the laptop at the work laptop that I literally never do any teleconferencing on, so I've learned that. That's not great. Okay.
Actually, while we are waiting, so maybe some people might have questions regarding the previous lecture. It would be a really good time to discuss.
There will also be a brief review at the beginning of this one, so you can stop me if there's something that didn't make sense in there.
Mm-hmm.
Yes, maybe I have one question.
Sure, yeah.
In the previous lecture, you told us about sequencing methods. And if I understood correctly, we break the sequence, and before copying, we attached adapts to the ends of the reads. Yes?
So I'm going to try to, I'm not sure I understood you. Let me try to repeat the question. You're saying that we shear up the sequence, we break the big jumps down, and then we add an adapter sequence of some known sequence to the ends?
Yes, I didn't quite understand why adapts are needed and what is it?
Yep, this is a great question, and I'm sorry I didn't do a better job explaining it. The reason you need the adapter sequences is that you're trying to do something called PCR. So that's polymerase chain reaction. And what you're doing when you do that is you're creating more copies of your sequence that you started with. But PCR requires something. It requires that you have a sequence of some known DNA sequence that you can hybridize a primer to. So a primer is a little 20 base pair or so segment of DNA that is the reverse complement of what you're trying to amplify. So if your sequence sits this way and you have an adapter at the end here, so it's going 5 to 3 this way, 5' to 3'. Your primer is the reverse complement of that adapter sequence and goes 5 to 3 this way. And what it will do is the polymerase will bind and it will make a copy of that molecule. The idea is that you want to make a lot more of your library, your DNA sequencing library or product, so you can make more of them. It's a big advantage if you know that at the end of this thing, basically all of the copies of DNA I have in here also have at their ends a sequence that I know the exact sequence that's in there. Because of at least for Illumina sequencing, the very way we identify these things and grow them on the slide, that flow cell, is based on binding that known sequence that we have in that adapter to the flow cell, which is just a bunch of these oligos that stick up like this. So it's kind of it's a necessary component of the way all Illumina sequencing works, basically.
I'm good, thank you.
Yeah, no worries. That's a great question. I wish I had done a better job explaining that the first time. So hopefully that worked this time. And like I said, I think a lot of you may not end up making your own sequencing data, but you probably will spend a good amount of time analyzing it, and it's wise to understand where it's coming from and how it got there. Cool. Before I get started, does anyone else have a question from last time? Okay. Vladimir, I guess I'll just get going if that's okay with you.
Yes, let's start then, Russ. So
right.
Yeah, you are very welcome to teach us today.
I think you need to set up my first account for screen sharing here, if you can.
Oh yeah, I need to stop sharing on my side.
Right,
yeah, sorry.
No worries, I got this. All right. Okay, we'll do desktop 1. All right, so hopefully, hey, I can even watch what I'm showing you. This is going to be great. Okay, so okay, welcome back. Today I'm going to tell you about genome assembly. So last time we talked about sequencing technologies and a little bit about where the data goes in QC. I know that I got cut off at the end So this time I've planned to go back and as a part of the demonstration I'll do with you guys today, we will go back and download data from the sequencing read archive so you can get some raw data onto your computer. We'll run it through some simple QC programs so you can see if the data is any good, and I'll kind of tell you how to interpret that. And then we will do some assembly as well. So we'll get to all of those things I promised last time. Okay. What just as an outline for today, there'll be a review at the first part of this. It should be relatively quick. Just what does variation in genomes look like? And I'll kind of emphasize the main kinds of variation that we care about in general in population genomics, and then a little bit about the kinds of variation that seems to matter a lot when you assemble genomes. We'll talk about the sequencing trade-offs and techniques. And we'll talk about, and I'll emphasize again, FASTQ format. That's probably the most important thing you can get out of this, because those quality scores, like I said last time, are going to be super important when we get to the genotyping portion of this class, and how to interpret genetic variation that Torfin will be doing, because it really is central to most of those calculations. I really think you should know it, and I'm going to emphasize it. Okay, from there, we're going to get into SRA toolkits, getting the SRA data. Actually, like I said, that's going to be a part of a demonstration. I'll just show you how you do that from the command line. Hopefully all of you has a Unix based system of some kind. In just a minute, I'll share a file that has some commands written out, but I'll also very, very slowly type those commands in, and you can follow along either way. After that, or before that, actually, this is out of order, don't worry. I'll talk a little bit about genome assembly for short reads. So the original ways people did this, why that doesn't seem to work as well as we would like it to, and then how people have been doing this sort of since then. And De Bruijn graphs are, or De Bruijn, I'm not sure, graphs are really commonly used for assembling most short read data. So metagenomics applications are mostly based on De Bruijn graphs, at least if you're doing any amount of de novo assembly. The original genome assemblies, or most genome assemblies from about 2009 through 2017, were all based entirely on Illumina, or mostly on Illumina. And so it's really been the main driver behind genome assembly. With that said, that is changing pretty quickly, and I won't go into the algorithms that people are using now for long read assembly, because they're changing so fast that I can't possibly tell you something that will be up to date, and you'll just have to learn it next time anyway. But I'll give you an idea what's going on. We'll talk a little about other ways people go about making genome assemblies and scaffolding their genomes, so linking libraries, things like that. And at the end, I will do an introduction to some of our work. So we've just published a paper earlier this year where we used a combination of technologies to solve an important problem in genome assembly. And I'm really excited to tell you guys about that, because it's sort of the most current and interesting thing I've got today. Okay, with that, let's get started. Okay, so we're going to start with a review, types of genetic variation. What I'm showing here are two of the key kinds of genetic variation that you will see used in population genomic inference. So the first is a single nucleotide polymorphism, or SNP. It's color-coded in red, SNP. And you can see that I've highlighted in red on the sequences here a SNP, right? So sample 1 has a C at this position, sample 2 has a G at this position. That would be a single nucleotide polymorphism if they're within the population. Okay. That's actually the bulk of all population genomics inference, is based entirely on the frequencies of these little things, SNPs. Another important kind of variation that you will see in a fair amount of population genomics, and you should be aware of, are indels. These are short missing bits of DNA. So in this case, CAC is present in sample 1, and sample 2 simply goes straight from GTC to ATT. So this is a short 3 base pair indel. It's another kind of variation that you see a lot of in this case. Indels are a type of duplication or deletion. But we kind of treat them separately than the way we treat macro deletions and duplications. So that would be deletions or duplications of very, very large segments of chromosomes, right? So you imagine here that we've zoomed way out, and I'm showing you all of chromosome 4, which is something like 180 megabases in humans. And it's deleted something like 20 megabases. This is a really, really big deletion as opposed to that 3 base pair indel. But these are tremendously important. The reason we treat them a little bit separately is that they are, you detect them in dramatically different ways, right? So that really short 3 base pair indel, a sequencing read, a short read, can just align over that and show you where the indel is. But when you're missing 20 megabases, it doesn't work quite the same way. You typically don't find those with straightforward short read alignment methods. And so there's a whole lot of specialized genomics approaches for going into these kinds of things. But there's increasing evidence that these are a really, really important part of phenotypic evolution and adaptation. And so I think it's really important to emphasize that gene duplications, deletions on these larger scales are an important part of genome structure variation. And in fact, you've probably heard the statistic that human and chimpanzees are somewhere between 1% and 2% divergent per site. That's only true in the alignable portion of their genome. They actually vary quite a lot more because of large-scale duplications and deletions where there simply is no alignable sequence between the human genome and the chimpanzee genome. So these are a really, really big component of evolutionary change. Okay. These are actually also a type of macro duplication and deletion. So that's to say that not all of these types of variation are fundamentally distinct from each other, but I emphasize them sort of throughout because they're very important in genome assembly, as we'll see today, and interpreting short read alignments to a reference genome, as I'll talk about in a week. So something to know about. Remember, again, just to remind you, transposons are little sections of DNA that are genomic parasites. They encode just a few proteins, just enough to basically cut themselves out, copy themselves, and reinsert themselves into the genome somewhere else, thereby increasing the number of copies in the genome and increasing their fitness because they're able to spread across the population this way. Okay. I'm not going to say too much about this. It's just a reminder that I find the UCSC genome browser or the Ensemble genome browser, and there's a few other specialized ones depending on what kind of data you're looking at. But I find these to be incredibly useful. And I know that data visualization is often sort of overlooked because people are, you think, oh, I don't need this. I'll just run my statistic and I'll look at the output. But it is often incredibly valuable. So if you identify a gene you think is experiencing selection, it's incredibly valuable to be able to go in, look at where it is in the genome, see what's around it and say, is this unusual or not? Is there something strange about this region? Are there reasons I shouldn't trust this? For example, if I saw a region where there were a ton of transposable elements, I might say, oh, my alignments aren't reliable in this region of the genome because that sequence has been copied so many places. It's hard to know if the reads that I've aligned there really come from there or somewhere else. And so that's happened to me before. Of course, we're getting better at flagging and removing those things, but it's still a possibility. And I do really recommend getting comfortable with some sort of data visualization platform so that you can look at these things. I'm not going to be able to have time to demonstrate this for this class, but I do want to emphasize also that it's very straightforward to just load up custom tracks. What that means is, each of these is a track here, right? These are your OMIM alleles, these are BRCA2 gene models, et cetera, and these are comparative genomics data to related mammal species. What I mean by this is that you could have your own track, right? So if you host this somewhere on a server, which I'm sure you can figure out how to do, you can load up the track in UCSC and actually compare a statistic you've computed against all the other information in here. So it allows you to really, really quickly visually cross-reference your information against everything else, which I find to be incredibly valuable in some cases. Cool. I'm not going to do a full review of sequencing technologies and how we actually make the data. For today, I'm going to emphasize the components of the data that seem to be really important, both for genome assembly and sequence read alignment and interpretation. So I'm going to contrast these two, and then I'll tell you about how this slide is already out of date, like all other slides in bioinformatics presentations. So on the left, we're talking about Illumina sequencing. You may remember this is the bulk of sequencing that was done between, say, 2008 or 9 and 2017, and it's still the bulk of sequencing today. The reason it's so appealing is that there's a very low error rate. Depending on how you do the math, you get something between 1 in 100 and 1 in 1,000 errors per site. So if your read's about 100 base pairs, the expected number of errors might be 1, but it might be less. And so it's a really appealing thing because you have extremely accurate short reads. The other very appealing feature of Illumina is that you get a lot of data. And so for a given amount of sequencing effort or money, you get a ton more data often, and it's really valuable. It often gives you something called paired end reads that we'll talk about a little bit later today and why those are super valuable. For now, just know that often you sequence from both ends of an input molecule, so you have some idea that 2 reads come from nearby in the genome. Okay. On the other side of that, we talked a little about longer read technologies, so Pacific Biosciences or PacBio and Oxford Nanopore. These tend to have higher error rates, and sort of the main trade-off for them is that you have error rates as high as maybe 1 in 10. It's going down all the time, but it's still typically higher than Illumina. But the really cool thing is you get long reads. So PacBio reads can be up to, say, 20 or 30 kilobases. But if you go all the way to Oxford Nanopore, people have reported reads as long as 2 megabases, so 2 million base pairs. And people routinely get reads as long as 100 KB all the time. You typically get fewer reads, so there's a trade-off for cost per number of reads. I want to emphasize one thing, which is that this is a little out of date. So earlier this year, PacBio released a new technology called Hifi, high fidelity long reads. And basically what that's done is made it so the error rate is down to Illumina levels, maybe 1 in 100, for reads that are maybe 10,000 base pairs to 20,000 base pairs. So a lot of the trade-off here is being taken away very quickly. It still costs a lot more to make that data relative to Illumina, but the appealing thing is having those long reads makes genome assembly almost trivial in some cases. It's getting kind of incredibly easy, and so I think it's Hifi is a very appealing thing going forward. And you will see a lot more of it, so I'm going to emphasize it now because when you read, you got to know what it is. Okay. There was a 2 pieces of terminology that you'll get. And I know we haven't actually aligned reads to a reference genome, so you kind of have to just take this as it is for now and know what I'm talking about when I say it, but we'll get to it in the next class. People often use these terms interchangeably, coverage, and because you'll see coverage in both of them, right? So they'll call the depth of coverage and breadth of coverage the same thing. They are not. That's not universally true. Many people make this distinction, but I want to, for the purposes of this class, just emphasize the simple terminology that I'll use, which is your depth of coverage is the average number of reads you stack up at a given site in your genome, right? So at this site here, there are 5 reads. You call your depth of coverage 5, versus your breadth of coverage, which would be the proportion of sites in the genome that have at least 1 read at them. So in this example, the vast majority of sites have at least 1 read. You might say these edges do not. So you would have maybe 95% of your sites have coverage. It's usually expressed as a fraction or a fraction. So you might say 95 out of 100. The important difference here is average number of reads, stack of reads is your depth of coverage. The proportion of your genome covered is your breadth of coverage. Okay. All right. The thing I said I want to emphasize a little bit more. FASTQ format. You're going to see this a lot. It's a really important component of everything that you do in bioinformatics. And so the way this works is it has a characteristic structure where each read or short sequence is represented as 4 lines. You have a unique sequence identifier. So this is typically a number corresponding to where on a flow cell the sequence came from. You have a sequence of DNA. There may sometimes be ends in here if there's very low quality bases, but usually it will just be these 4 characters, A, C, T, and G. You have a line that you can skip. It has to start with a plus, but then it can have anything else in it and it doesn't matter. And then finally, you have the quality string. This is ASCII encoded FRED64 normally. It can also be FRED32 in some cases. And there's, you can just look it up as you go. So I skipped a slide here. We'll come back to this right now. This is just a very simple math. It's stoichiometry, but I think it's worth knowing because you may be asked to design an experiment someday, which is how to compute your informal expected depth of coverage. And the simple thing to do is usually to multiply the number of reads by the base pairs per read, divide by your genome size, and that is your expected depth of coverage. Of course, you can also move these things around a little bit, right? So if you reformulate this equation and you want to know the number of reads that you need to sequence to get a depth of coverage, you could simply multiply your genome size by your target depth, divide by the reads that you get per read pair or molecule, and that would be your number of required reads. Okay, cool. Jumping back again to FASTQ, often this will come as paired end reads. Usually what that means is you'll get two separate files that have very, very similar sequence IDs at each line and sequences in each file. They usually won't have the exact same quality string. I am being lazy and made that up for today, but usually they will have exactly the same number of lines in file 1 as in file 2, because every read that has a first read has a paired read. And so you would have two files that you feed it, and I'll explain how to do that when we get to the assembly portion in class. Okay. So this is a really short exercise I'm putting in here. I'm not even going to send people to breakout rooms. What I just want you to do is think a little bit about this sequence here, right? So up on the side here, I'm showing you this read, and it has this sequence and this quality string. And what I'm showing you are two candidate alignments. So they're regions of the genome that look a lot like that read, except each of them has one difference. What I want you to do is figure out which of these alignments should be preferred and why, reminding you that this is your Fred scaled probability, that there is an error at a given site. And I don't want to give away too much, so I'll just say that much for now, and you can figure this out. And I'll be right back. I'll give you all another minute, unless somebody thinks they already have it. Hopefully it's not too bad. Okay, in the interest of not calling on people I don't know randomly, I'm just going to tell you all right now what I think the right answer to this question is, and we can talk about it if anybody's not sure. The simple intuition for this is these alignments are nearly the same. There's just one position that I've actually bolded for you in each that doesn't match the reference sequence. So all you need to do really is to figure out which of these two is more likely to be an error in the sequencing read than the other. And that basically tells you which alignment ought to be preferred because it's more likely to match the sequence correctly. So what you do is you go to this A over here in your quality string, and you see that that corresponds to ASCII G. We go down to this table, we look at ASCII G, we see, oh wow, the probability of an error there is very, very low, right? You get Q38 and a probability of an error of 0.00016. So very small number, very likely that that's a correct sequence here, this A. In contrast, if we go to this G, it's fourth from the end in the ASCII string. That would be this exclamation point. That's sort of the opposite case. ASCII 33, probability error 1. So we're pretty darn sure this G is wrong. In that case, the alignment that we should prefer is the second one. The reason is that if this G is very likely to be wrong, then there's an even better chance this read actually really does match the reference here. Whereas if this A in the read is almost certainly right, there's almost certainly a real difference relative to the reference sequence at this site. So we would typically prefer the alignment that minimizes the probability that there's a real difference between the read and the reference. This isn't necessarily how we do short read alignment. It's more an example to get you thinking about ASCII encoded quality scores, but it's, I think, also useful just for thinking about this a little bit. Okay, cool. This was a bit of review as well. The SRA database, which we will interface on the command line with today, can be found and browsed online if you need it. So again, reminder from last time, this might be useful. One thing I like to emphasize is you may sometimes be given raw data. Rather than back it up on servers that you maintain and pay for, it is very cheap and easy to simply upload it to the SRA immediately. And then the SRA will do the backup for you because it maintains that sequence. And in fact, you don't even have to release the data publicly for quite a while after uploading it. So this can be a really appealing thing. And I actually do this almost always as soon as I get raw data, is just upload immediately to the SRA. Cool. I'm skipping a lot of slides today. Okay. So a lot of genomics is based on a k-mer. This is going to be something new. And what I mean by a k-mer is a sequence of length k that's usually a subset of a total sequence. So in this case, what we've done is we've taken this sequence in black at the top, which is this here, and we've found all sequences, all 6 mers within that, right? So A, G, T, T, G, A would be the first 6 mer, it's the first 6 bases. If you go over 1, the next 6 mer would be G, T, T, G, A, G. The reason this is really appealing is that you can then find, even if 2 reads don't exactly overlap, you can discover very, very quickly if they share a substring, because they might overlap for a k-mer's distance or more. And if they do, you've got a very good chance of discovering that overlap using this kind of approach. Okay, often what you'll do with raw data is look at your k-mer distribution. So what that means is I take all my raw data in, they're sort of represented up here as black reads, and I'll pare them down to k-mers. This might be reads, sequences of length 31 or something like that. It's usually reasonably short, but long enough that it's fairly unique in the genome. If you then count all of those up and plot the frequencies of the k-mers in your genome here, you'll see a couple things. In red, you'll see errors, basically. So errors should be relatively uncommon if they're all independent and random across your reads. For example, here you see that these 4 have this error, but everything else has a different sequence at that site. That would be over here somewhere in the k-mer distribution because it should be relatively uncommon or even completely absent from any other read. In contrast, if you go out here to what we call the main peak in this diagram, what that corresponds to is the depth of your genome, basically. So in this case, your depth is about 35x. And we know that because if most of my unique k-mers have a depth somewhere in the range of 35 or a median depth around 35, it strongly suggests that that's the actual sequencing depth of my genome. That's about how many reads I would have per site, assuming most of the genome contains unique sequence, which is mostly true. There is one other secondary peak that is actually really interesting here, which is down here. In orange, this is what you'd call your heterozygous alleles, or your diploid peak. You'll see that it's almost exactly half the depth of your main peak, which is what we would expect, right? So what's happening there is that all of my reads randomly sample one of 2 chromosomes in an individual. That's diploid, that has an allele at a site that doesn't match the other chromosome. Half the reads match the reference allele, half the reads match the alternate allele. And as a consequence, you get 2 different k-mers with that difference in them at almost exactly half the depth of the main peak. Okay. There's a few other things in here, so they're calling them repeat humps. I don't really know why, but the short answer is repeats can extend the number of k-mers quite a bit. Remember, I talked about transposable elements. That's one of the main forms of repeats. There are many others, and those can be extremely abundant. So this isn't even really doing this justice. You often see k-mers way out here on your graph in the, depths are thousands of k-mers range relative to your main peak. So the reason I bring this up is it's a really useful way to learn a lot about your genome. Even before you do any amount of genome assembly, you can just take your raw reads and learn something about it very quickly. I also bring it up because k-mers are sort of an integral component to the way that people do De Bruijn graph-based assembly and that kind of thing, and I'll show you that in just a second here. Okay, so the main topic of today, the genome assembly problem. So genomes are long, and they're complex strings of A's, C's, G's, and T's. Basically, that's it. It's a 4-letter alphabet, and it spells out, many millions of base pair words, if you will. most sequencing data is actually very short, right? So it's hundreds of base pairs. In some cases, it's longer, as we described. Hifi and Oxford nanopore reads are getting longer, but for now, just assume it's relatively short. And of course, there's a limit to how long reads can possibly be because DNA extraction isn't perfect. You always shear your DNA at least a little bit when you do it. And so I don't think anytime soon we'll be sequencing from all the way from one telomere to the other telomere, all the way across a chromosome. But of course, that would be the goal at some point in this process. For now, the way this works is we have some unknown genome that we're trying to assemble. But what we actually have are a bunch of very short sequences, right? So the analogy people often use for this is that you start with some large, complex report, some written out report on many, many pages, and then you shred those pages. So you turn them into tiny little strips of paper. And then you're trying to figure out how all that paper goes back together to tell the story of that long, complex report. And I think you can imagine that that would be a tremendously frustrating problem. It has been frustrating for a lot of people, but it's also been one of the most active areas of research in bioinformatics, and it's been really one of the main drivers behind the growth of population genomics is the fact that we can get these genome sequences increasingly well from many, many different species to study kind of anything we think is interesting. Okay, so your typical genome assembly steps would look something like this, and we'll go through all of these today, but we'll spend the vast majority of the time on the primary assembly. The primary assembly is the act of putting these short read sequences together into what we call contigs. So contigs are sections of, and that's short for contiguous. So it's sections of DNA that we're pretty darn sure about based on the pairwise alignments or structure of our short read data that we've aligned to each other. You often then do something called scaffolding, which is finding links between contigs, and you can do this with either those paired end reads I was talking about, larger linking libraries that I'll talk about later. You can do it with comparison to related species. So in some cases, you know the genome sequence of a close relative, and you might say, well, the gene order is probably pretty similar between, say, human and chimpanzee genomes. I can probably use chimpanzee to see where the human scaffold contigs go along this genome. You can use other sources of information like recombination maps. There's actually a bunch of different ways to do this. But the basic idea is to lay your contigs out such that their orientation relative to each other represents a chromosome, even if you don't fill in all of the gaps in between them. And then finally, you usually do a step called polishing, which is to correct small or large errors in your primary assembly that you've just made. Okay, cool. So when genome sequencing first became a big thing, at the time we had 1,000-ish base pair reads that were very, very accurate, but we didn't have many of them. So there was a very appealing way to assemble those. It's called overlap layout consensus. The idea is you find the overlaps between all pairs of sequences. So you literally compare every single sequence to every other sequence. Then what you do is you lay them out based on those comparisons. So you're basically constructing a graph where each node in your graph is one of these reads, and each edge in the graph is an overlap between reads. So we've decided some threshold on what overlap is reliable, and we leave that in here. In some cases, you can actually weight those edges by what you think the reliability of the overlap is, or by how similar the sequences are. But often you just have an unweighted edge in your sequence. This is directional, right? So it's a comparison between, say, this chunk and this chunk, not just anywhere on this read and anywhere else on this read. Okay. There's also things I will not talk about today, which is like simplifying this graph structure quite a bit. Often there are many, many paths through a graph that are otherwise essentially identical because all of your sequences overlap some amount. So you go from, if you have 3 sequences that overlap like this, and you go from here to here to here, or those two overlap a little bit and you just go directly, that's actually the exact same path. So there's lots of simplification that goes into this to make a much smaller graph. And finally, at the end of that whole thing, you try to find a Hamiltonian path, which means you visit each of these nodes exactly once, and you take the consensus. Okay. The consensus is just, if I stack up all my reads at a site, what's the most common base at that site? Okay. But this didn't scale, right? So I said this is the way we did it originally, but it actually wasn't the way that the vast majority of genome sequences that are available today were made. And the reason for that is that the overlapping step is an enormous computational bottleneck. So what you're doing in that step, as I said earlier, but I'm going to repeat for emphasis, is you're literally comparing every single read to every other read. And if you don't have a lot of data, that's not a big problem. But one of the main issues with Illumina was we suddenly had an absolute ton of data, right? So it wasn't very hard to make many, many millions of reads, and to do all those comparisons can be very expensive. And just to spell it out for you one time, if you have n reads, then there's approximately n squared pairwise comparisons you're going to do. So for example, if you have 400 million reads, you're going to have 1.6 times 10 to the 17th comparisons. So this isn't something you're going to be able to do very easily, even with extremely optimized ways of computing the overlap between sequences. It's just a very expensive problem. And so the real bottleneck here is not that the algorithm is a bad one. Overlap layout consensus is great. It's just that it's extremely computationally expensive once you have an absolute ton of data or an absolute ton of depth at a given site. Okay. So the solution that most people came to, these remain extremely popular today. In fact, there's an assembler, a very famous assembler from, I believe, St. Petersburg, called the St. Petersburg assembler. Correct me if that's not right. But it's often called Spades as well, and it does all kinds of fun stuff. The originally was a De Broglie graph assembler, and I think it still mostly is. So what these can do is you reduce your computational load. And the reason it does that is it simplifies the heck out of your overlapping problem, which is to say, I don't have to do all pairwise comparisons anymore. So if you want to make a De Broglie graph, we'll start by just showing you how you make a De Broglie graph from a single read. And the way you do that is you identify all of your k-mers within a given read. So if my read is the sequence at the top here, ATC, AAG, CG. And I go through it, and I'm trying to find all k-mers of length 3. The first one would be ATC. Then you would have TCA, CAA, AAG, AGC, GCG, right? In other words, I found all possible 3 base pair sequences across this whole thing. Okay, if I want to make the graph, what I'll do is I'll treat each of these k-mers as an edge in the graph. And then each of my nodes is a k-1 mer on the left. So in other words, for GCG, GC is your k-1 mer here. And CG is your k-1 mer here on the right. And so you can do the same thing again for another sequencing read, right? So just to emphasize the point here, we imagine here we have 2 reads, and in this case, we've colored the overlap between them as this CG here. This goes CG, CGT, GTA, etc. I won't read them all to you right now. But again, it's the same basic thing. You'll have your k-1 mers represented as your nodes. And so if we looked at what those graphs look like across the entire reads, in this case, I've moved sequencing read 2 up here. And now what we're doing is we're showing you the De Broglie graph across the whole read. What you can see is the way you read this is each of these edges, again, is a k-mer of length 3. And each of these nodes is the k-1 mer on either side of that k-mer. Okay. You'll notice there's a really important difference between these graphs. The first is great. It's linear. It has this very nice shape where it just reads from one end to the other across the sequence. In fact, you can read the sequence off of this if you simply read the first letter of each of these k-mers as you go across, right? A, T, C, etc. This one is not so pretty. It has a lot going on, but you can still find a single path through this that will visit all of the nodes at least once if you go across this, right? So it would go GC, GT, CG, GT, etc. this way, back up to GT, over and down. But you can see it's got this loop in it, which is a little ugly. Okay. You then merge those graphs, right? So the way genome assembly works, this is the big reason De Broglie graphs are so appealing, is that now we're looking at which reads visit those exact same nodes and edges, have passed through the same nodes and edges, right? So if I combine those two, you see that they do overlap, right? At this little segment here, and then a few other places as well, just by chance. But importantly, it overlaps at that CG where it should truly overlap. And so you can use this to find overlaps between your reads really, really quickly, because we didn't do an all-by-all comparison. All we had to do was figure out which k-mers were in the reads and then figure out which reads had the same k-mers in them by putting them onto our graph. Okay, so you can see how that's really appealing, right? It reduces this overlap problem quite a lot because now you're just looking at a very, very small search space of these shorter k-mer sequences relative to all possible combinations between reads. Okay, so there's some important trade-offs when you do this kind of thing. One of them is the k-mer size. So everyone that learns about these asks the question of, what's the right k-mer length? And the answer is, I don't know for sure. There's been a lot of work in this, and I won't go into it in great detail. I'm just going to give you the high-level principle here, which is usually if you have higher k-mer values, you have higher specificity. That is, if you do find an overlap between two sequences, you can be increasingly sure that that overlap is unique, and those two reads actually go together at a given site. In this case, let's imagine we used k equals 4, or 5, sorry. What we've done is deconvolute our reads into our k-mers, and you see ATCAA, GCTAA, right? They're all k-mers of length 5 now. If we lay them out like this, remember that each side has the k-1 mer relative to that edge. There's a problem, which is they don't actually visit the same node in this graph. So we can't put these two reads together because the overlap between them is just this CG, right? So you have higher specificity, but you have lower sensitivity. It's much, much harder to see those overlaps because you need increasingly long overlaps in order to see which reads go together. To show you the other end of that spectrum really quickly, let's look at a more degenerate case. So in this case, you're looking at k-mers of length 2, which means your k-1 mers have length 1. Or in other words, the only possible nodes in my graph are now A, T, C, and G. So this has become a problem. And in fact, when we try to visit, it'll be very, very hard to figure out what the correct path through this is just using this size k-mer. So what you end up getting usually is an assembly that breaks at many of these loops and is a really, really short assembly. So the big trade-off here is you have higher sensitivity with lower k-mers, but you have much lower specificity. So we found a lot of overlaps. It's just that none of these overlaps are reliable and we can't use this information. Usually this trade-off is also related to sequencing depth. So if you sequence really, really deeply, that is, you have many, many reads at every site in the genome, it's often good to turn up your k value. So use a higher k-mer. The reason is because you sequence so deeply, you're much more likely to have some number of reads that overlap at the same position, even if k is a relatively long sequence relative to the read length itself. Obviously, you cannot make a k-mer graph with a k-mer value longer than the read length. So there's an upper limit for sure, but it's useful to turn that up in some cases and see how this goes. And we'll also do that today in the exercise. Okay, cool. So a slightly more complete example, since those are sort of toy examples. If we imagine that we have these 5 sequencing reads here, they are 1, 2, 3, 4, 5. We've actually already aligned them on the correct sequence down here, but you wouldn't know that in advance, of course. You have to learn that by doing this. And we then figure out what all of the k-mers of length 7 are. So you have your k-1 mers of length 6 here. What you can see is that the first read supports this set of chunks here, 4 goes across these. And so when I lay these reads on top of each other, there's sufficient overlap in all cases, right? Each of these has at least one k-1 mer of length 6 overlap between the next adjacent read, such that you're able to lay them out correctly across this and reconstruct the whole sequence. So at the end, you come up with ATGG, blah, 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 by just reading across this, right? The ATG is the furthest out here, AATC over here. Hopefully this is making some sense. You get to puzzle through this in just a minute. Remember, the first step will always just be finding the k-mers in your sequencing reads and then figuring out how they go together. Okay. To tell you some of the challenges of genome assembly, one of the main challenges that was encountered very quickly early on was bubbles. So a bubble in your graph is a case like this, where there's an allelic variant. In this case, it's shown as A and B. So we imagine this is a diploid individual that has 4 reads at this site. And 3 of them have an A and 1 has the B. So this is going ATC, TCA, CAA, AAG, AGC, etc. But because the other reed has a B, it diverges at this point, right? It doesn't go from ATC to TCA, it goes from ATC to TCB. And actually the graph is separate. This is called a bubble. You can see how it's a little plopped out part of your Kamer graph. And it's something that happens quite commonly in certain species. And in fact, if you imagine a case where your species has so many mutations in its genome that it's heterozygous at a site, say every 10 base pairs, then if you try to reconstruct its genome with K equals some large value, say 51, your 2 graphs would be completely separate from each other because they would always encounter a new variant that meant they never got merged. And you'd actually assemble both of your chromosomes completely independently from one another. So bubbles can be really important for this reason. It's hard to know when you've encountered one. And in most, I think all, but at least most De Bruijn graph-based programs go through and do a step called bubble popping. So they look for things like this, and they usually select the path that has the most support. So in this case, you'd say 3 reeds went up, 1 went down. We're going to select the path that goes through A, not through B. Okay. Another really important case is something called tips. Now I'm not even showing you the sequences. Just imagine this is a zoomed out view of your De Bruijn graph where there's some anonymous node and edge going across this thing. If you had a case where there was an error at the very last base in a reed, you'd get something like this, where the final Kamer doesn't match the final Kamer this way. But hopefully what you would find, if you lined everything up, is that your edge weights supported this linear progression across your path, and you remove the tip as well. So tips and bubbles are the 2 main graph artifacts you see when doing this kind of thing. And the vast majority of software that's been written has lots and lots of steps to remove these sorts of problems. Okay. As I said earlier, just to give you a more concrete example, we use what's called an edge weight to choose between alternate paths. The edge weight is simply the number of times a given Kamer was seen, right? So in this case, TAC has edge weight 2, whereas down here, TTC has edge weight 1. In other words, we've counted the number of reads that progress this way through my graph versus this way through the graph. And that's usually sufficient to identify the one that's more likely, right? Especially if these are errors. As I said earlier, errors should be really, really uncommon, and those edges should be very poorly supported. Okay, and to demonstrate that again here, you can see that if we had our edge weights, we imagine that we have 9 or 10 reads stretching across this section here, my graph, and just one that diverges up and has these and has this spurious edge out here. In this case, it's slightly different than the one I showed you before, where you would probably have an error in the second to last position in your read, because there are 2 unique Kamers until the end of this tip. But again, tips tend to have low weight, and we remove them. Okay. So I know I've talked about repeats way too much in this class already, but this is the first time they come up in a way that actually affects what we're doing. Is the De Bruijn graph always is the Euler graph? Yes, so the difference is in overlap layout consensus, you're trying to find your Hamilton path that visits all the nodes. In a De Bruijn graph, you want to visit all your edges using, yes, it's a Euler path. That's a great question, by the way. So those of you that are familiar with string theory type things, look it up. It's great. It can give you a little bit more intuition for what we're doing here. What's going to happen here is I'll explain a little bit about how they break. So for example, you've got these 2 green sections can be considered repeats. That is, it has the exact same sequence twice. I'm sorry, I missed a question earlier. Let me try to answer this. Why is the quality string there are fewer characters than in a read reference? If I did that, it's because I didn't do a good job creating the example. The quality string should always have the exact same length as the read in your sequence. The reference won't have a quality string necessarily. It's often just going to be in what's called FASTA format, which means we don't try to represent the possibilities of errors in the reference. That's not always true. You sometimes get them in FASTQ format, but it's much more common to see a reference in FASTA format, which simply has the sequence, and that's it, and we assume it's true. Okay, back to this example. We've got, sorry, lost my place. In yellow, you have a unique sequence. In light blue, you have a unique sequence. In red, you have a unique sequence. We know they're unique because they only have one path through the graph. You don't re-enter it somehow via this cycle here. In this case, you do, right? So you go across your yellow unique sequence, you enter the green sequence, and that can exit in two different directions, right? It can exit in either pink or back to light blue, which goes back to green. In this case, you might be able to kind of figure it out that the only way to completely traverse this graph from end to end is to go yellow, green, blue, green, red. But it's not always going to be true. A lot of these cycles are impossible to break. And the reason they can be impossible to break is that if that repeat is longer than a given read or a k-mer value, you can't see from end to end across it. It kind of just stops your graph at some point and you can't keep going. Okay. One thing we'll talk about really quickly is one of the main summaries people use for their assemblies, and then we'll do an in-class exercise and an example. So one of the primary, perhaps the primary statistic that's used to summarize a genome assembly is what's called an N50. And so if at the end of constructing my genome, I had 9 total contigs with lengths 2, 3, 4, 5, 6, blah, 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 up to 10, what we want to do is figure out the N50, which is the fewest number of contigs whose lengths sum to at least half of the total distance, right? So if you added all this up, you'd get 54. That's the total length of my assembly. What I want to know is what's the fewest number of contigs whose length sum to half of this. And in this case, it's 8 plus 9 plus 10 equals 27. So your N50 would be the longest contig above which length half of your assembly is contained, right? So in this case, 8, 9, and 10, the last 3 contigs actually contain half of the total assembly. And so when we define this as a single statistic, your N50 is now 8 base pairs. I'm going to try to say that slightly differently because I know that it can be a little bit confusing. The N50 is a simple way of summarizing the total contiguity of your assembly, right? So your genome assembly has some lengths for each of the contigs, right? In this case, I took a very easy 2 through 10. The N50 is the size of contig that if you took contigs of that size and every larger contig in your genome, they would sum to at least half of the length of your genome assembly. So of the total length of your genome assembly. So in this case, it's 8 because again, the contigs of length 8, 9, and 10 are a total of 27 bases. 27 divided by 54 is a half. So it's exactly your N50. It rounds up. So for example, if this was actually a 7 and you had 26 out of 54, it would not be a half, and you would actually be 1 lower. You'd have 5 included as well. This is a really quick and straightforward way of summarizing genome assemblies, and it's extremely widely used. So that's why I emphasize it here. Your N50 is probably the first thing you'll look at when you make a new genome assembly because it tells you something about just how well you put everything together. It does not mean that you joined everything correctly. And I think that there are many cases where it can be misleading to use the N50 because there's a lot of false joins in your assembly of sequences that don't actually go together. But it's a useful first approximation to how good a job we did. And as I said earlier, your optimal K value is often very hard to know in advance. And the solution that most people have come to for this is to just try a bunch of different K values when they construct their De Bruijn graph-based assembly. So I'm showing you an example from probably data I simulated. But the important point is, if you go across this and we look at our N50, so we summarize this assembly. And in this case, the total length of the assembly should be about 1,600 base pairs. There's exactly 1 contig in this assembly, so you've done great. Down here at K equals 15, it's apparently quite hard to resolve all the loops and spurious connections you find. Remember, your sensitivity is very high when K values are low, but your specificity is very low. So there's many, many loops in your De Bruijn graph. The program's not able to resolve those. And as a consequence, you only have very, very short contigs that come out of this and a small N50. As you go along this, you see it's going up. And this is that trade-off I'm describing is at play here. Your specificity is going up, your sensitivity goes down. But presumably because the read depth is relatively high, when K is equal to about 31, it does really well. Interestingly, as you go to k equals 35, it's clearly lost its sensitivity, and is so much harder than crappy academic code that I usually write. So I know this game. Okay, let's bring them back. I don't want to leave them out there for too long. Close all rooms. There we go. And I'll go back to my screen share so you can see this. Okay. Okay. Are they back? No, still waiting. Give them a minute. A couple are back. Okay. Many people are back. Does anyone feel like they got the answers to these two questions before I randomly call on people and force you to answer me? Does anybody feel like they got one of these and wants to share? All right, let's look at our list of participants over here. How about Dmitry Biba? Can you tell me?
Okay. Well, I've constructed the graphs for most of sequences, but I haven't thought about them yet.
Okay. Why don't we do it together? I'll tell you about it a little bit. I don't have the actual graphs drawn out, so I'm going to have to sort of describe this to you guys. But if you were constructing this graph with k equals 4, or rather k equals 5, let's start there because it doesn't work. If you had k equals 5, then the last 5 mer in this first read would be C, G, G, T, A, right? So it's k minus 1 mer that lives on the left would be C, G, G, T. And so for these two, for this read to be linked to any other read would require that they overlap those 5 bases because they have to have that same node and that same edge between the node in order to be in the same graph. Otherwise, you wouldn't see this. And so in this case, you have GGTA. You see that that overlaps these 4 bases here, but it doesn't overlap all the way to the C. So if you used k equals 5, you couldn't possibly assemble the sequence because you wouldn't get all of these onto the same graph. But if you use k equals 4, you would be okay because your k minus 1 mer would be GGT on the left here. And the k minus 1 mer of this last k mer of 4 would be GGT. So they'd overlap at that node, they'd have the same edge, and they'd overlap at the right node, and those would be on the same graph. So the answer to the first question, what's the largest possible k mer you can use to get this sequence, would be 4, using a De Bruijn graph. Obviously, you could do other things to get this solved. Okay. All right, what's the smallest possible k mer that you can use to assemble this graph without loops? And what you basically want to do then is think about are there any repeats in this sequence? And I believe there are, right? So there's this AT here and this AT here. And so if k was equal to 2, obviously there'd be loops because all of your k equal 1 mers would be A, C, G, and T. There have to be loops when you do that. That's not a valid way to do this really, although it is formally possible. And so what's going to actually happen here is that the smallest possible would be 3 because you want to be 1 larger than that minimum edge length or the minimum node distance AT here. Otherwise, you have to revisit that same node at least twice and you'll have a loop in your graph. That doesn't mean you couldn't construct it. Remember, this is a directed graph and there's still probably one straightforward way to get all the way through it. So you might still be able to assemble this, but you would probably have a loop in there. Does that kind of make sense?
Yes.
Okay, cool. So that's the way to think about these. I'll emphasize before we go on that there is an absolute ton of different software packages for doing this sort of thing. But in general, those packages have slightly different approaches for different problems. Today, we're going to use one called Velvet, which I'll introduce as we do the exercise. So just to give you an overview of what this exercise will be, we're going to download data using an SRA toolkit, which is a command line utility for interfacing with the SRA. We're going to check the data quality using FastQC. We'll assemble the genome using Velvet, and if there's time, a bonus section where we use BLAST to figure out what this genome actually is. And so before I do any of that, I'm going to give you guys a file in the chat function here. And give me 1 second to find it for you. Okay. Okay, so in your chat, you should all see a file. It should be named assembly_example.txt. Do you all have that? Does one of you have that? Okay. Dimitri, do you have that?
I experience some problems with Zoom right now.
Oh, okay. I'll go really slowly. Hopefully most of you can see this. It has annotated commands that I'm going to run today, and hopefully will make some sense to you guys as we go.
Yeah, right. I think you're not showing the screen.
Right. I'm not showing the screen right now. I'm talking about the file I've transmitted to you guys.
Oh, sorry.
So we'll go back to sharing in just a second here. Hopefully you have the file. If you don't, do your best to follow along as I type the commands. Okay, back to sharing. Share. So hopefully you can see my whole screen. We're going to do this, and we're going to open up my example so I can follow my own stuff. Okay, and we'll close that and close that. Okay, so here we are. Let's close this too. It's going to annoy me. Okay, so we're going to admit somebody from the waiting room. Okay. The assembly example is here. You can see both these screens, hopefully, and it's clear enough if your monitors are big enough. And if not, I'll try to just say what I'm doing. So the first thing we're going to do is get some things installed, right? So I asked last time, you all have Conda. So Conda install is how you do this. Everything we're going to do is in the BioConda channel. So see BioConda. And first we'll do SRA tools, okay? I probably already have this, so it's probably not going to do anything. Some of you may actually be installing this, though, if you haven't already. Right, so I already have the packages. Your output may be slightly different than mine, in large part because you don't already have this. So hopefully you're all getting the thing installed. Please let me know via the chat if you're not, or ping Vladimir and he can help you as well. Okay, so we're going to get that. We're also going to get FastQC. Thankfully, all of these things are available via Conda, so it should be very easy to get these installed on pretty much any Unix platform. I think you can even do this on Windows now, but I'm not sure because I never use Windows. Okay, and then finally the package Velvet. So Velvet is a De Bruijn graph assembler. It's actually broken down into 2 steps, but it is basically what I've been talking about. It does this, and it's great. Okay. So let's make sure these things actually installed on our system. So first we'll use the command fastqdump. Most packages in bioinformatics will give you a help statement if you haven't given it enough information to do something useful. So in this case, I type fastqdump, and it tells me how I actually should have used the program if I knew what I was doing. Right, so let's do that now. We're gonna first download some reads we want. And I know that they're paired ends, so I'm going to type fastqdump, and then I'm going to type this command --split--files. In other words, it will split the paired end reads because SRA entries for paired reads are 1 entry. We'll gzip the output just for the sake of good practices. It doesn't really matter. This is a very small file, but I think it's still useful. And the file is going to be SRR 10504 875. Okay, what that is an accession number, the short read archive accession number for this read group, SRR 105048875. So let's try that. It takes about a second and it gives you this output, right? So it read 18,800 spots and it wrote 18,800 spots, right? So it did pretty good. Okay, so let's take a look at this. If I hit LS now, I should have these two files, right? So I've downloaded and split my files. You see two FASTQ files. That's because these are, as I said, paired end reads, which means they will have the exact same number of lines in them. So we can use zcat, which will just uncat to the screen, and we can count the number of lines in the file like that. What I'm doing there is I'm decompressing the file, piping it through standard out. That's what that straight up character means. And I'm piping that into a utility that's basically built into all Unix systems called word count. In this case, I'm telling it -L. In other words, tell me how many lines did you see in this file. And if we do the same thing for its pair, right? So it's the same command, except instead of _1, I'm doing it for read file _2. So it's paired file. And I do this again. I see that they are the exact same length. So 75,200. That isn't exactly equal to the number of reads in the file. And the reason is, to emphasize, your FASTQ format has 4 lines per read. So actually it would be 75,200 divided by 4 reads in each of these files, which I'm gonna bet you is equal to 18,800 number of spots we wrote, which would make some sense. Okay, let's actually look at one of these so we see what FASTQ format looks like in this case. We'll look at the first 8 lines, which should correspond to the first 2 reads, right? And if we do that, we see this, where this first line is your sequence identifier. It tells you something about it. In this case, these were actually aligned as well, although you're not to know that yet, which is why it tells you what reference it was aligned to and where on the reference it was aligned. But also it tells you the length of the read. So these are all 50 base pair reads, which was a common length for a while. As I said, there's this line that you can basically ignore here as well that starts with a plus and then has actually the exact same information as in your sequence identifier line. I don't know why we bother with that, but it's there, ignore it. Then you get your quality string. Because this is a good FASTQ file and not one that I wrote out for this exercise poorly, it has the exact same number of characters as the sequence read up here, right? So each of these corresponds to the probability that the base above is correct. In a lot of genome assembly, that's actually ignored. I'm pointing this out again because it will matter a lot when we get to the interpreting genetic variation section of this class. Often in genome assembly, they just ignore the quality string altogether and just look at the sequence. And I think Velvet does that as well, although I should check, I'm not sure. Okay, so what are we gonna do? The first thing we're gonna do is we're gonna run this command, fastqc, and we'll do it on each of these files, right? So you can actually just give it both and it will run. It actually does them separately though. So you see the output very quickly. It's analyzed each of these 2 files. And if we hit ls again, we'll see what it's created. And in this case, we'll do this. And in this case, what you can see here is it's got a zip folder containing the actual output. And then it actually creates an HTML page for each of these 2 files, which is a little bit annoying if you're trying to do this on a server. But if you're doing it on your own laptop, it's incredibly valuable because now I'm just gonna go to this section, this directory that I'm in now. And if I click on one of these 2, my computer will figure out that I want to open an HTML file and it'll just go to the web browser and open it. And you're now able to very easily visualize your FastQC report. So the second thing I think you should do, the minute you get your FASTQ reads from your sequencer, you should back them up to the SRA. But the second thing you should do is run FastQC, or there's a couple other packages, but I'm going to teach you this one. FastQC, and just really quickly go through it and make sure your sequence is any good. And in this case, it kind of tells you what you should worry about. So most things do fine in this sequence. It does have an issue with the per base and the per sequence quality scores, especially the per base quality score right here. And the reason that it's concerned about this is that the quality is not actually that high for Illumina data, even at the beginning of your reads. But if you get to the end, remember, this is what it's showing you here is the position in the read. Illumina sequencing goes in cycles. So the first base that was added to each of these clusters was actually reasonably high quality. Your median's up here at like 29. But by the time you get to the end of your reads, the 50th cycle, your median quality is down here at something like 18 or 19. So it gets a lot worse really, really quickly. Okay, so that's great. But that actually didn't quite demonstrate what I wanted to. We're going to use these sequencing runs in just a second. In the meantime, what I'm going to do is download a run that I know is a bad run that I want to use as an example to show you what should happen with FastQC when you have crummy data, okay? So again, we're doing the same thing. We're gonna say SR FASTQ dump, right? And for those of you following along on the document I sent, we should be right here, basically. Going back to that. So FASTQ dump, these are actually single end reads, and I know that already. So you don't need the split files option. Okay, so let's download that. And that's hopefully gonna happen for us. It's taking a second longer than it usually does. Ideally, my internet is connected. Okay, we got that. So let's hit ls again. As I said, these are single end reads, so they don't have a pair, so there's no _1 or 2 after them. But let's run fastqc again. So fastqc, and this time we'll just give it 107. And if any of you are relatively new to Unix and wondering how I'm typing so fast, the answer is that I'm not. I'm using the tab to autocomplete. So if you hit your tab key and there's only one possible file name you could be typing out, your computer will figure this out for you and will put it down at the end here. So if I hit fastqc real quick, it'll run through the analysis. It's finished. Okay. And so if we do this again, ls ltrah, what we can see is that we have now created some new files, right? So we had this FASTQ data we downloaded. There's again this zip folder that contains all your real results, but is extremely challenging to parse. And then there's this HTML page, which will be the kind of summary of all the results that you're really trying to get to in the first place. So if we go over to that, in this case, it's this one here, and we load that up. Come on. There we go. We load it up. As I said earlier, this is an example of a bad run. How do I know that? Well, if you look over here, you'll see that there's a per base sequence quality, sequence content issue, right? If my reads were completely random across this genome, they should have the exact same rate of A's, C's, G's, and T's at each position in the read. The fact that these don't is mildly concerning, but it's not that unusual for a transposase. That's why it says warning. If we go down here a little bit more, we'll see something much more concerning. So in this case, this is your GC profile for the real data, right? In other words, it's the percent GC in each of your reads. And then in blue is the theoretical distribution that you would get if these were if G's and C's were more or less equally distributed across these reads, which is basically true in most genomes. The fact that you see two completely distinct peaks here should be very concerning, and it suggests that there's contamination, or that at a minimum, you actually have two different genomes in this sample rather than one genome, as you would expect. Okay, but there's something even more concerning. So if we go down, you'll say, oh, a lot of sequences are duplicated, right? So we actually see that we have many, many sequences where we see the same sequence more than once. And this happens when you do that PCR step I talked about just way too many times relative to how many molecules were in your library originally, such that you're actually sequencing the same molecules or descendants of the same molecules many, many times over. That's bad. And then finally at the end, the really concerning thing here is your adapter content, right? So what's happening here is it's showing you across each of the cycles in these reads, and this read goes up to 150 base pairs, that's why it's all the way out here. You'll see that by around base 35, we start seeing a lot of what's called a Nextera transposase index sequence. In other words, these reads have a ton of the adapter sequence inserted in them. So they're not even, the sequence isn't all from the genome, it's actually a lot from your adapters as well. That can always be resolved by trimming, but that's the real value of running fastqc quickly, is it tells you what you need to do to make your sequence usable or not. All right, that's not the main point of today, but I think it's really valuable. We're gonna head back down. I'm gonna show you today, which is over here, and I don't need that anymore, and we'll do this. Okay, so we're back to this screen. The real point of this though, so that's how you run fastqc, but the real point of this is to run a program called Velvet. And Velvet is one of the very first de Bruijn graph assemblers that was made. It has been cited something like 10,000 times and is an incredibly popular way to do these things. There are many others and many more specialized approaches for this sort of thing out there, so I'm not necessarily suggesting that Velvet is the catch-all solution to an assembly problem, but it's pretty easy to use and it just generally makes some sense. Velvet works in two steps. The first is to create your hashes of some specified k-mer length. So we're just going to call our directory assembly. You tell it, so let's make sure Velvet's assembled. Velvet H is the first command. There you go. You can see that you have quite a lot of detailed usage here. For our purposes, I'll tell you what commands to run, but you can always go back here and see all kinds of fun, different ways to run this. So in this case, we'll run Velvet H. Again, that's the one that creates the hash. It's the first part of this program suite. Short paired. So we have short paired end sequences. The format is fastq.gz, so they're gzipped fastq, so you tell it that as well. And then we're just going to let it do this. So one. Okay, that's the command you need to run for now. All that's going to do is create the hash table for these two paired end reads with k-mers equal to 21. And it's going to create a directory called assembly where the information it needs is stored to run the actual graph building and consensus calling portion, which is the second program in this suite. Okay, that was quick. So Velvet H is done. Let's run Velvet G. So Velvet G, again, is the section that builds our graph. There are many, many parameters for this as well. We're not going to go into any of them today. You're just going to run the defaults. But basically, you give it the name of the assembly that holds your hash that you created with Velvet H, and you run it again. Okay, and now we've done that, and it gave us a bunch of log as it was running. It's an extremely verbose program, and it tells you all sorts of fun things. Like, for example, it's removing null nodes, it removes tips, all kinds of cool stuff is going on here. But then at the very end, it also tells us the things we really want to know, which is how good was our assembly. For example, this one has a final graph with 262 nodes. That's after combining lots and lots of things. An N50 of 374 and a maximum content length of 952. So it isn't that long, but it's reasonably long. So it did pretty well. Okay, but really, like I said at the beginning, probably what you'll want to do is run a bunch of different k-mer values in order to get the real output, get a real idea what your assembly looks like and identify an optimal k-mer value, right? So like I said, it depends on your coverage, it depends on your complexity, lots of things. It's actually hard to know. What most people do, and what I certainly recommend, is you just try a couple of different k-mers and see what happens. And Velvet's typically pretty fast, so this will work. You can run a bunch of k-mers quickly. Okay, so Velvet H, assembly, 11, 42, 10. Okay, and I'll explain what this is in just a second. Okay, so what I'm doing now is I'm asking Velvet to create the hashes starting at k equals 11, all the way up to k equals 42 is what I want to use, 42, and in steps of 10. In other words, this will create 11, 21, 31, and 41. So if I do that, it's going to take a second to do them all. Okay, it's done. That was quick. And then let's see what it made. Now you can see it's made the same sort of directory, right? Like we made this first one the first time. Now it's made a hash table in each of these directories, but it's also told me the k value it used to do that. So 11, 21, 31, and 41. Now you can't actually run Velvet G on each of these by any straightforward command, but you can use some simple bash scripting to do this. If you're in a different kind of shell like TCH or something, I don't know how to help you, just run it separately. But if you're in bash, and most of you probably are, for I in 11, 21, 31, 41, right? We'll say do Velvet G assembly. So what I'm doing now is I'm telling this program to run for each of this variable I've created I in all the k values I created, 11, 21, 31, 41. Do this assembly command. And that's it. So it's going to run them all in succession. It may take a second to do them all. And this time, instead of just writing my log to the screen in this horrible long format, I'm going to store it in a file so I can parse it more easily later. So each of them will be stored in a unique file, in this case, log_k, where I, again, corresponds to 11, 21, 31, and 41. We're done. Let's take a look at that. You'll see it looks the same, except now it's also created these log files. There's a very simple way to get the answer I want. which is if I do this, if I cat all those files together, and then I grep out a line that contains this statistic I care about, the N50. And remember, the N50 is a way of quickly summarizing how continuous my assembly is, where a larger number would typically be good. I want to get as much of my genome into one molecule as possible. We do that, you get something like this, where you have a final graph, where you see your summary of your final graph in each of these cases. And in this case, there's an absolute ton of nodes. You have an N50 of 4 and a max of 34. So your actual maximum context size is shorter than your read length, which is 50. This is a very, very bad graph. It's an example of what happens when you give a relatively small k value, because you just had a ton of loops across the entire graph. It was unresolvable. And Velvet just said, I'm not going to even try. I'm shutting this down. Okay. You do a little better at k equals 21. So these are displayed in order, right? The first one corresponds to k equals 11, this line here. The next line corresponds to k equals 21 here. And in this case, you do a little better, right? You have an N50 of 277, so at least it's longer than the read length of 50. We put stuff together. And there's a max of 809, so it's not terrible, but it's not great. If we then go down a little more, you see we actually did quite well at k equals 31. You got a max of 7,991, and that's pretty good, right? An N50 of 3,738. So your molecule size for which your assembly contains molecules of that length or longer for half of all base pairs is 3,738. So this is a pretty continuous assembly. And then finally, you can see what happens when you have too many, or when k value is a little too high, so 41. Your specificity is too high, there aren't enough overlaps between your reads, and we don't get a very good assembly out of this either, right? So we have a max of 343, and even more concerningly, we actually are assembling many fewer reads into sequences. So the total assembly length is 10,000, which is probably not right. Okay, cool. That's the basic outline of how k values work. Hopefully that showed you that what I was talking about in the presentation is more or less correct. Now what we're going to do is the same sort of thing again, only this time, let's demonstrate what happens when we have fewer reads, right? So remember what I said is there's a trade-off. Your k value is related to sensitivity and specificity. But if you have many fewer reads, then you should have many fewer overlaps between your reads, and you'll need a smaller k value in order to put stuff together. So we're going to do that, and if you're following along in the document, you can do that over here, right? So what I'm doing is subsampling my reads. Remember, there were 75,000-ish lines originally. Now we're going to knock that number down. And we're going to knock it down by just taking the first 25,000. We'll pipe that into gzip, so we still have a gzip file at the end. And you need to call that something different because, of course, you'll be overwriting your file otherwise. So you see, I'm calling it almost the same thing, but I'm using .subset to indicate to myself that this is a subset of the total reads. So if you run that, you actually need to run the same thing for the second set of reads here. So exact same command, zcat that first, take the first 25,000 lines, gzip it, output it. We have our subsets now. So let's do the same thing again, right? So we're going to use Velvet H to create our hash structure in the first place. 4210. Okay, fastq.gz. And we're going to do something lazy. So that's a wildcard. What that means is match anything that looks like this. And in this case, there's only two files that have the extension .subset.fastq. So it will match the correct files and it will run them rather than typing them all the way out. That's what that little asterisk is doing for me, right there. Okay, so let's try that. Okay, we've created all our tables. It went even faster because there's many fewer reads. So now we'll do the same thing again, only we're going to assemble, right? So for I in 11, actually, let's show you that this exists. So we've created all of these directories now containing those hashes. All right, so for I in 11, 21, 31, 41, do Velvet G assembly reduced. Reduced. Okay, And we're going to try to make the assembly out of each of these using that command there. It's done very quickly, but just to reiterate what I'm doing, for those of you trying to follow along, I'm using this new variable I that I'm declaring here, and I'm using it at all the hash values I use, so 11, 21, 31, 41. I'm running that exact same assembly command on each of them just by default, and I'm storing the log as log reduced k with that variable I gave it here as the file name. Okay, if we look at all that, you see I've now created all those log files. So let's look at them the exact same way as we did for the logs of the original assembly. If we do that and we grep on N50, this will go in alphanumeric order, which will be in order. So k equals 11, 21, 31, and 41. And we'll get something like this. And what you can see is that our answer is slightly different. Just to remind you, I'll do the first one now. So this is what we got when we used the full read set. And this is what happened when we used this reduced read set. And what you can see is that probably a better assembly is produced with fewer reads when k equals 21, or at least a more continuous assembly, than when k equals 31 here. So in this case, what we had was probably that there were so many overlaps that you saw some loops between sequences that shouldn't have been there. And so when k equals 21 here, it didn't work that well. But when you had many fewer reads, finding those extra overlaps by having a shorter k-mer is extremely valuable, and you find many more connections. And so your graph can be more contiguous. And in this case, what you see here is that we have an N50 of 2,356 and a maximum contig length of 4,702. So it did really well. If you go down here, you'll see that again, it's K31, whereas the K21 here had a N50 of 277. It did actually really poorly. So the point of this is to say that I'm using literally the same input data, just a subset of it, about a third of the input data, and it dramatically affects the performance. So it really is valuable to mess with that K value, and it really affects the way we do this. Okay, I have a little bit more of a presentation to get through today, and I'm probably not going to get to everything. But that's the basic example. Those of you that like that file I sent has lots and lots of information for you about how I'm running these commands and why. So it's annotated throughout, so you can run it yourself. And in fact, you can just run the whole thing by typing bash and then the name of the file, and it will do the whole thing for you. So give that a shot, see what happens. In the meantime, I'll go back and finish up this presentation. Okay. So we talked about that. One thing I want to emphasize is that long read assemblies resolve many of these problems. So in this case, what you've got here are these R's, right? And what that's meant to indicate is that they're a repeat. And when your short read assembly has a read length that's smaller than your repeat length, or much smaller than your repeat length, it can be very challenging to know how this all goes together. So the approximate graph that comes out is something like this: A into R, C back to R, B back to R. And it's impossible to know which order these should go in. It can go A, R, C, R, B, R, or it could go the other way, A, R, B, R, C, R. And so you've got a problem there. But with long reads, the real reason that they're becoming so popular is you can just read right across that repeat, right? This went all the way from the blue stuff to the yellow stuff. And so we can be sure it goes A to R to B to R to C to R. That's what's going on right now. That's why this is so popular and interesting. Okay. One thing that you should know is that most long read assemblers, not all, but many have returned to this idea of overlap layout consensus. And the big reason is there's so many errors. You have one error in 10 or so in a long read that you just don't ever get long overlapping k-mers, because you're just so likely to have an error in a given k-mer, unless it's extremely short and therefore not useful, that you just have this issue built in where you need your k-mers to be unique, but unique in the genome sequence, but because they're so long, they're very likely to contain an error, which means you won't identify exact overlaps. Overlap layout consensus is appealing because you will spend time optimizing all those pairwise alignments, and you can be pretty sure that there's a good alignment there, even if there's a high error rate, as long as you have a long segment that aligns between your reads. Okay. One more thing I'll talk about that you should be aware of because it's becoming extremely popular. I mentioned this last time, but I'm going to re-emphasize it today, is what's called chromatin conformation capture. And this is a way of doing that step I described at the beginning called scaffolding. And what you're trying to do here is take all of your contigs from across your genome and figure out what order they go in along a chromosome, and figure out which ones are on the same chromosome and different chromosomes. And the way this works is you start with your genome. And your genome is actually folded up on itself a ton of times, and it's maintained with this protein complex that kind of holds it all together. And then what you can do is cross-link all those proteins so that the three-dimensional conformation of the genome is more or less fixed in place. That's the second step here. You can then shear your DNA. There's a bunch of ways to do this. In this case, they're describing something called omni-seq. It doesn't matter. You shear the DNA, but the protein complex stays in place and holds everything more or less together. And then you actually ligate those DNA molecules to each other. And sometimes they back ligate. They just find the exact same original DNA that it was cut from. But in many cases, it finds something else. And in that case, it found a sequence that was nearby in three-dimensional space of the genome, so very likely on the same chromosome, but it wasn't right next to the molecule originally. And so these libraries can span big distances between read pairs. You get up to a couple megabases, millions of base pairs apart. And it's one of the biggest ways that people have started succeeding in putting together larger complex genomes, even without resolving all those repeat structures we talked about. Okay. And what this looks like is something like this. Where what we're showing you is the all-by-all comparison, where if one read aligns to this point of the chromosome, it's showing you where the pair aligns, and then where you see darker red colors, there are many, many read pairs that are near each other. What you can see is that across all of chromosome 1, this is a mosquito example, across all of chromosome 1, there are many, many contacts that are very near each other along the chromosome. But those are spanning pretty big distances. This is about 40 megabase chromosomes, so the distance across here is something like a megabase or 2. But nonetheless, most of your signal is in the on diagonal. That is, the reads are relatively nearby to its pair in the linear space of the genome, but they still span big distances. So it can be really valuable for seeing those connections. In this case, the genome was already assembled, so it's not too hard to use this to do well. And you can see that one other really cool feature of this comes out, which is that the ends of your chromosomes, your telomeres, tend to be touching, right? So you can see that the end of chromosome 2 and the end of chromosome 1 must be touching. You have an excess of contacts here. The centromeres are often touching their telomeres. You can see an excess of contacts here. I won't go into the biology of why, but the cool thing about this is it both reveals real biology and can be used for genome assembly. Okay. So what's happened recently is we're moving away from what we call draft assemblies. So almost any genome that was made between about 2009 and 2016, 2017 would be called a draft assembly by today's standards. That's not universally true, but the vast majority. And the reason is because they were just a pile of contigs. They didn't get to this scaffolding step. So they just used Illumina data and they made thousands and thousands of contigs and had no idea where along the chromosome they were. Today, it's becoming much less common and much more common to make relatively complete assemblies where you have this nice linking information and can put the whole genome together. Okay. Often when you do a genome assembly, you're actually using many different technologies. So what you're going to do is use a combination of short reads. Remember, they're accurate and they're cheap, but they're short. You'll use longer reads, which are inaccurate but long, and maybe a linking library as well, because you're able to span enormous distances and figure out how your contigs are arranged along the genome. We're going to skip that today. Just be aware that there is a giant database on NCBI that has tons and tons of genomes, and you could search through them and find literally hundreds of thousands of reference genomes. It's getting pretty impressive what's out there. Okay. What I want to end this is just an example from our recent work to kind of show you how this works in practice and tell you a little bit about how you can combine multiple technologies to do this sort of thing. I also just like to talk about myself, so hopefully that's not so bad. Okay, so we started with a problem. And the problem we started with is, let's imagine that you could only get a single individual from a species, from a very small species. So in this case, we use Drosophila melanogaster, which already has a very nice genome assembly. But that's really nice for us because we know the right answer. So when we develop a method to assemble a genome, we can compare back to that and show that it basically worked. The other challenge of these is it's mostly a molecular challenge, which is they're tiny. You can only get a couple hundred nanograms of DNA out of them, which is very, very small and certainly not enough to make an Oxford nanopore library, a usual Oxford nanopore library. We made some kind of special ones. And so there's a big challenge just with the scale of what you're working on. If you really only had one individual, how would you do this? And so we set up this cross. I crossed 2 different strains of Drosophila to produce a completely heterozygous outbred individual. The idea is to make this conservatively challenging so that we're dealing with something that you really would find in nature. And then we did two things. We first extracted from the back half of the fly, so we used the abdomen to do this. We extract high molecular weight DNA, and we make what's a pretty specialized ONT sequencing library. I won't go into the details, but the thing to know is that it's more accurate because we use molecular tricks to read the same molecule many times over. We make an assembly out of those, and then simultaneously we take a little bit of DNA, a tiny amount, and we make a library out of that by fragmenting and adding our adapter sequences, right? So these adapters are used in PCR. And then we make an Illumina paired-end library out of that, and we assemble that with a De Bruyne graph-based assembler called Miraculous. And Miraculous is very similar to Velvet. The one big thing it does is it knows about heterozygosity, and it's actually trying to figure out where those bubbles in your graph are and use those bubbles to assemble the two diploid chromosomes by identifying which mutations are near each other. So this works really well for extremely heterozygous species like Drosophila. Okay, while we did all that, the second half of this experiment is to do Hi-C scaffolding. So we took the front half of the fly and we made a Hi-C library. So we cross-linked all the chromatin. We did our restriction enzyme digest. We add our adapter sequences using biotin to pull them down, and then we do PCR to enrich for a good library. And that also goes into Illumina paired-end sequencing. And what happened, we merge our two primary assemblies here. And then finally, we run this program called HiRISE to scaffold out our assembly and figure out how it all goes together using the information from chromatin conformation capture. At the very end, we do what's called polishing. And to do that, we just realigned all of our short-read DNA sequences and figured out what the most common variant at a given site was and called that the correct answer. And we do that 4 times because there's lots and lots of little errors in your primary assembly that you can iteratively get out by running these polishing programs many times over. Okay. To show you that what I'm describing was all real, here's kind of what happens. When you run Miraculous, so that's using just your short Illumina data, we got to a contig N50 of 51 kb, which is actually pretty good for a species like this, and an assembly size of 112 megabases, which is also about right. It's a little shorter than the true assembly, and the reason is we're missing something, which I'll describe in a minute. We also use a different program, WTBG2, to assemble the long read data we produced, and you get a contig N50 of about 100 kb doing that, and actually a very, very similar total assembly size, so that's good. And the nice thing is when we merge those two, you see that some of the stuff the long reads span the short reads couldn't, and probably some of the stuff the short reads span the long reads couldn't. But in combination, you get a merged assembly that's actually much longer. It's 274 kilobases. So it's actually become a pretty decent assembly. And as you might expect, it's about the same size. It's a tiny bit smaller because there's sections that aren't confirmed by the other and vice versa. Finally, we do our scaffolding step here. We're using HiRISE to do this. There's nothing you need to know about that other than it is a scaffolding program. And that's the big difference. It goes from a 274 kilobase N50 to a scaffold N50 of 26 million base pairs. In other words, these are now chromosome scale assemblies for this species. So the idea is HiRISE, by using that linking library, is able to put it all together into chromosomes. We do some polishing, et cetera. It gets a little longer. And then finally, we remove a bacterium from here, and you end up with a genome of about 111 megabases, as you might expect. As I said, it's missing something, right? The real Drosophila genome is somewhere near 138 megabases, maybe a little bigger. So what's wrong? Well, what's wrong, actually, before I show you that here, what's wrong is this. If I compare this assembly to the Drosophila melanogaster assembly, you'll see that what we're missing are mostly what I talked about, those mobile genetic elements or transposable elements that we've been talking about this whole time, these repeats that you see all over the genome. Our assembly did a really poor job of discovering those. We only got 55% of the correct ones. tRNAs are also fairly repetitive. They're often in big arrays with the same sequence in them, more or less. And in that case, we only got 76% of them. Whereas the rest of this, so genes, exons, long non-coding RNA, et cetera, we actually do quite well. We get about 95%. We're quite happy with those outputs. So it suggests that that really is the big limitation, as we said before, that if your reads aren't quite long enough, you can't really discover these repetitive elements in many cases. One thing I do like to point out, I have this a little out of order, is that if we compare the two, this is what's called a dot plot, where you see a dot that connects the two assemblies. So ours is this high-rise scaffolded assembly here, and this is the Drosophila melanogaster reference genome. If you have this nice one-to-one correspondence across it, it means that you've pretty much assembled the exact same genome. Right? You have this nice linear relationship where one sequence in DM6 corresponds to one site in the high-rise scaffold, and we've arranged them from end to end, such that you see something like this. So this would be considered a very, very high-quality genome. And in fact, when we went back and looked at how many misassemblies we had only about 800 total misassemblies in the whole thing. So we did quite well to do this. Okay. So your conclusion here is that the combination of many technologies is a really useful way for challenging genome assembly. Repeat elements are, as I described, a big issue for primary assemblies and even not resolved with some semi-long reads in some cases, so it can still be a challenge. Okay.